Exercise is one of the most powerful and well-studied ways to stimulate mitochondrial biogenesis, the process by which cells generate new mitochondria, thereby increasing energy production capacity. When muscles and other tissues are subjected to physical stress, the demand for ATP, the energy currency of the cell, rises significantly. This increased demand signals the body to adapt by producing additional mitochondria, enhancing the ability of cells to generate energy efficiently. Both high-intensity interval training, HIIT, and resistance training are particularly effective because they create acute, intense energy demands that trigger robust cellular responses. Endurance exercise, on the other hand, promotes mitochondrial density and function over longer periods, allowing cells to sustain aerobic energy production during prolonged activity. At the cellular level, exercise activates key signaling pathways that drive mitochondrial biogenesis. One of the most important is the PGC1A pathway, which regulates the transcription of genes responsible for mitochondrial replication and function. During exercise, stress signals such as increased calcium levels, AMP-Chi-ATP ratio changes, and reactive oxygen species production activate PGC1A. This activation initiates a cascade that increases the number of mitochondria in cells, improves their efficiency, and enhances their capacity to produce ATP. Over time, repeated activation of this pathway through consistent exercise leads to a measurable increase in both mitochondrial quantity and quality, directly translating into improved endurance, strength, and overall energy availability. Different types of exercise stress the mitochondria in distinct ways, producing complementary adaptations. High-intensity interval training challenges anaerobic and aerobic energy systems, prompting rapid ATP production and stimulating both fast-twitch and slow-twitch muscle fibers. This form of exercise creates strong bursts of metabolic stress, which are particularly effective at signaling the need for new mitochondria. Resistance training, while typically associated with muscular hypertrophy, also increases mitochondrial density in the working muscles, supporting improved energy supply for repeated contractions. Endurance exercise, such as running, cycling, or swimming, predominantly stimulates oxidative metabolism, increasing the number of mitochondria in slow-twitch muscle fibers and enhancing the efficiency of oxygen utilization. By combining different forms of exercise, individuals can maximize mitochondrial adaptations across multiple tissue types and energy systems. Exercise also improves the functional quality of mitochondria. Beyond increasing the number of mitochondria, physical activity enhances the efficiency of existing mitochondria, optimizing ATP production, and reducing the generation of damaging reactive oxygen species. This is achieved through improvements in mitochondrial dynamics, including fusion and fission processes, which maintain mitochondrial integrity and remove damaged organelles. Improved mitochondrial function allows cells to sustain higher energy output with less metabolic strain, supporting both physical performance and long-term cellular health. The benefits of exercise-induced mitochondrial biogenesis extend beyond muscle tissue. Organs with high energy demands, including the heart and brain, also experience enhanced mitochondrial function with regular exercise. In the brain, increased mitochondrial efficiency supports cognitive performance, neuroplasticity, and resilience to stress. In the heart, enhanced mitochondrial function improves cardiovascular efficiency, endurance, and recovery from physical exertion. Collectively, these adaptations contribute to greater overall energy, improved physiological resilience, and protection against age-related decline in mitochondrial function. By consistently engaging in targeted physical activity, including high-intensity interval training, endurance exercise, and resistance training, the body stimulates mitochondrial biogenesis and enhances energy production across multiple tissues. These adaptations increase cellular efficiency, improve endurance and strength, and support overall energy levels and long-term health. Through regular and strategically designed exercise, mitochondria become more numerous, efficient, and resilient, providing the foundation for sustained energy, improved performance, and long-term vitality. This process strengthens not only muscles, but also key organs like the heart and brain, supporting overall health and longevity at a cellular level. Nutrition plays a central role in optimizing mitochondrial function, providing the essential building blocks and cofactors that mitochondria require to produce energy efficiently. Mitochondria rely on a variety of vitamins, minerals, and antioxidants to carry out the complex biochemical reactions that convert macronutrients into ATP, the energy currency of the cell. Without sufficient nutrient support, mitochondrial energy production can become inefficient, leading to fatigue, reduced performance, and accelerated cellular aging. By focusing on nutrient-dense foods and strategic dietary patterns, individuals can enhance mitochondrial efficiency, support cellular resilience, and promote long-term energy and health. B vitamins are among the most critical nutrients for mitochondrial function. Vitamins such as B1, thiamine, B2, riboflavin, B3, niacin, B5, pantothenic acid, and B12, cobalamin, serve as cofactors in the biochemical pathways that convert carbohydrates, fats, and proteins into ATP. For example, riboflavin is a component of flavin adenine dinucleotide FAD, which plays a key role in the electron transport chain, while niacin is part of NAD+, a central molecule in energy transfer and redox reactions. Adequate intake of these vitamins ensures that mitochondria can efficiently process energy substrates, minimize the production of reactive oxygen species, and maintain cellular homeostasis. Minerals also support mitochondrial energy production and enzymatic function. Magnesium is required for ATP synthesis and stability, acting as a cofactor for numerous enzymatic reactions within the mitochondria. Iron is essential for the function of cytochromes, which are critical components of the electron transport chain that generate ATP through oxidative phosphorylation. Selenium and zinc contribute to antioxidant defenses that protect mitochondria from oxidative damage. A diet rich in these minerals, obtained through whole foods like leafy greens, nuts, seeds, legumes, and lean meats, supports optimal mitochondrial performance and resilience. Antioxidants play a dual role in mitochondrial health. They neutralize harmful reactive oxygen species while supporting adaptive cellular signaling. While mitochondria naturally produce reactive oxygen species as a byproduct of energy production, excessive oxidative stress can damage mitochondrial membranes, DNA, and proteins, impairing function. Antioxidant-rich foods such as berries, colorful vegetables, and certain spices provide polyphenols and other compounds that reduce oxidative damage while promoting mitochondrial adaptation and repair. These compounds help maintain mitochondrial integrity, prevent premature aging, and enhance energy efficiency at the cellular level. Dietary patterns can further influence mitochondrial function. Intermittent fasting, time-restricted eating, or controlled periods of reduced caloric intake stimulate cellular stress responses that enhance mitochondrial biogenesis, repair, and efficiency. These approaches trigger adaptive pathways, including AMPK and sirtuin activation, which improve mitochondrial energy production and reduce metabolic strain. Combining nutrient-dense meals with strategic timing optimizes both the raw materials for mitochondrial activity and the signaling pathways that drive adaptation. Healthy fats, particularly omega-3 fatty acids, support mitochondrial membrane integrity and function. The lipid composition of mitochondrial membranes influences the efficiency of electron transport and ATP synthesis. Consuming sources of omega-3s, such as fatty fish, flax seeds, and walnuts, helps maintain membrane fluidity, supports efficient energy transfer, and enhances resistance to oxidative damage. Balanced macronutrient intake ensures mitochondria receive both the substrates and structural components required for optimal function. By providing essential vitamins, minerals, antioxidants, and healthy fats, nutrition directly supports mitochondrial energy production, resilience, and repair. Adequate nutrient intake enables mitochondria to efficiently convert energy, minimize oxidative stress, and maintain cellular homeostasis. Through nutrient-dense dietary choices, strategic timing, and attention to essential cofactors, mitochondrial function can be optimized, resulting in improved cellular energy, enhanced resilience, and long-term health. Proper nutrition creates the foundation for sustained vitality, cognitive clarity, and overall physiological efficiency, supporting both energy levels and longevity. Sleep and circadian alignment are critical regulators of mitochondrial health, playing a central role in energy production, cellular repair, and long-term physiological resilience. Mitochondria are dynamic organelles that respond not only to physical activity and nutrition, but also to the body's internal clock. During sleep, especially in deep, slow-wave stages, cells engage in repair processes that restore mitochondrial integrity, remove damaged organelles, and optimize energy production for the following day. Disruptions in sleep timing duration, or quality can impair these processes, reducing mitochondrial efficiency, increasing oxidative stress, and accelerating cellular aging. By prioritizing consistent sleep schedules and alignment with natural circadian rhythms, mitochondrial function can be preserved and enhanced, supporting both daily energy and long-term health. Circadian rhythms, the body's internal 24-hour clock, regulate numerous physiological processes, including mitochondrial activity. Many mitochondrial enzymes and energy-related pathways fluctuate according to circadian signals, optimizing energy production during periods of activity and promoting repair during rest. When sleep is misaligned, such as with irregular schedules, shift work, or exposure to artificial light at night, these rhythms become disrupted. Mitochondria may produce energy less efficiently, generate more reactive oxygen species, and fail to engage in repair processes at the appropriate times. Over time, chronic circadian misalignment can contribute to fatigue, metabolic dysregulation, and increased risk of age-related disease. During deep sleep, mitochondrial maintenance processes are particularly active. Cells use this period to engage in mitophagy, the selective removal of damaged mitochondria, and mitochondrial biogenesis, the production of new organelles. Mitophagy is essential because damaged mitochondria generate excessive reactive oxygen species, which can damage DNA, proteins, and lipids. Removing these dysfunctional mitochondria and replacing them with new, efficient organelles ensures optimal energy production and cellular resilience. Adequate sleep provides the uninterrupted time necessary for these processes to occur effectively, reinforcing the link between rest and mitochondrial health. Sleep also affects the hormonal environment that influences mitochondrial function. Hormones such as melatonin and growth hormone peak during sleep and play crucial roles in regulating energy metabolism and cellular repair. Melatonin, beyond regulating circadian rhythms, acts as a powerful antioxidant within mitochondria, protecting them from oxidative stress and supporting energy production. Growth hormone stimulates tissue repair, enhances protein synthesis, and indirectly supports mitochondrial efficiency. Disrupted sleep reduces the secretion of these hormones, impairing mitochondrial maintenance and compromising energy levels during waking hours. Light exposure and daily routines further influence mitochondrial health through circadian regulation. Exposure to natural sunlight in the morning helps synchronize circadian rhythms, optimizing mitochondrial energy production during active periods. Conversely, minimizing exposure to bright artificial light in the evening helps signal the body to transition into rest, supporting the mitochondrial repair processes that occur during sleep. Maintaining a consistent sleep-wake schedule reinforces these rhythms, allowing mitochondria to operate at peak efficiency and reducing metabolic stress. By aligning sleep patterns with natural circadian rhythms, the body creates an environment where mitochondrial repair, biogenesis, and energy optimization can occur efficiently. Proper sleep duration and timing not only enhance immediate energy levels but also protect mitochondria from age-related decline and oxidative damage. Prioritizing high-quality sleep and maintaining circadian alignment ensures that mitochondria can perform essential repair and energy production functions. Over time, these practices enhance cellular efficiency, support metabolic health, reduce oxidative stress, and promote both immediate vitality and long-term resilience, laying a foundation for sustained energy, cognitive clarity, and overall longevity. Deliberate exposure to hormetic stressors, such as heat, cold, or intermittent fasting, has a profound impact on mitochondrial function and resilience. Hormesis refers to the principle that small, manageable stressors activate adaptive cellular responses that improve overall function and resistance to future stress. When mitochondria are exposed to these controlled challenges, they respond by enhancing their efficiency, increasing their number, and bolstering antioxidant defenses. This adaptive response strengthens cellular energy production, reduces oxidative damage, and supports long-term physiological health. By strategically incorporating hormetic stressors into daily life, individuals can improve both immediate energy availability and long-term mitochondrial performance. Heat exposure, such as sauna sessions, stimulates mitochondria by increasing metabolic demand and activating protective cellular pathways. Elevated temperatures induce mild cellular stress, prompting mitochondria to produce more ATP and upregulate heat shock proteins, which repair damaged proteins and maintain mitochondrial integrity. This process enhances mitochondrial efficiency, improves energy output, and reduces the accumulation of reactive oxygen species. Over time, regular heat exposure trains mitochondria to operate more effectively under both thermal and metabolic stress, supporting cardiovascular health, endurance, and recovery. Cold exposure is another powerful hormetic stimulus for mitochondria. Brief exposure to cold, such as cold showers, ice baths, or cold weather activity, triggers adaptive responses that increase mitochondrial density and improve metabolic flexibility. The cold activates pathways that enhance fat oxidation, stimulate brown adipose tissue, and promote thermogenesis, all of which rely on mitochondrial activity. By repeatedly challenging the body with controlled cold stress, mitochondria adapt to optimize energy production under varying conditions, improving resilience and supporting overall metabolic health. Intermittent fasting or time-restricted eating also serves as a hormetic stressor for mitochondria. Periods of caloric restriction activate cellular energy sensors, such as AMPK and sirtuins, which stimulate mitochondrial biogenesis and enhance oxidative efficiency. These stress-activated pathways encourage mitochondria to operate more efficiently, generate energy with less waste, and reduce oxidative damage. Additionally, intermittent fasting promotes autophagy, a cellular cleaning process that removes dysfunctional mitochondria, replacing them with new, efficient organelles. This cyclical stress and repair process strengthens mitochondrial function and supports energy balance, metabolic health, and longevity. Hormetic stressors also improve mitochondrial resilience in response to oxidative challenges. Mitochondria naturally produce reactive oxygen species during energy production, and excessive oxidative stress can impair their function. Controlled exposure to heat, cold, or fasting induces adaptive antioxidant responses within mitochondria, enhancing their ability to neutralize free radicals. This increases mitochondrial efficiency and reduces long-term cellular damage, contributing to healthy aging and sustained energy levels. Integrating multiple forms of hormetic stress amplifies mitochondrial benefits. Combining physical activity with environmental stressors, such as post-exercise cold exposure or fasting before a workout, further enhances mitochondrial adaptations. These combined stimuli create a robust signaling environment that encourages biogenesis, improves efficiency, and strengthens energy metabolism. The key is consistency and gradual adaptation, allowing mitochondria to respond effectively without overwhelming the system. By incorporating controlled hormetic stressors, mitochondria are challenged in ways that stimulate adaptive responses, improve efficiency, and enhance resilience. Heat, cold, and fasting each trigger pathways that increase mitochondrial number, optimize energy production, and bolster antioxidant defenses. Through regular structured exposure to hormetic stress, mitochondrial function becomes more efficient, adaptable, and resilient. These adaptations support higher energy levels, better metabolic performance, and long-term cellular health, laying the foundation for vitality, longevity, and optimal physiological function. Mitochondrial health is closely linked to cognitive performance, mental clarity, and overall brain energy. The brain is one of the most energy-demanding organs in the body, consuming roughly 20% of the body's total energy, despite representing only about 2% of body weight. Efficient mitochondrial function is essential to meet this demand, as neurons rely heavily on ATP for synaptic transmission, ion channel function, and the maintenance of resting membrane potential. When mitochondria are operating at peak efficiency, the brain has the energy required to support attention, memory, problem-solving, and emotional regulation. Conversely, impaired mitochondrial function can lead to mental fatigue, reduced cognitive processing speed, mood disturbances, and increased susceptibility to neurodegenerative conditions. One key mechanism by which mitochondria support brain function is through ATP production. Neurons communicate through electrochemical signals, which require constant energy to propagate. ATP generated by mitochondria fuels sodium-potassium pumps, which maintain the electrochemical gradients necessary for action potentials. Without adequate mitochondrial energy, neuronal firing becomes less efficient, slowing information processing and reducing cognitive performance. By enhancing mitochondrial density and function in neurons, the brain is better equipped to sustain high levels of activity, focus, and mental endurance throughout the day. Mitochondria also influence cognitive function by regulating oxidative stress. The brain is particularly vulnerable to reactive oxygen species because of its high oxygen consumption and abundant lipid content. While some reactive oxygen species play a role in signaling and adaptation, Excessive production can damage mitochondrial DNA, proteins, and membranes, impairing energy production. Supporting mitochondrial health through lifestyle factors such as exercise, proper nutrition, sleep, and hormetic stressors enhances antioxidant defenses, reducing oxidative damage and preserving neural function. Efficient mitochondria produce energy with minimal waste, allowing neurons to operate optimally while maintaining long-term resilience. Hormetic stressors, including physical exercise, intermittent fasting, and controlled heat or cold exposure, further enhance mitochondrial capacity in the brain. Exercise increases cerebral blood flow, delivering oxygen and nutrients that support mitochondrial function. Additionally, exercise stimulates the production of neurotrophic factors such as BDNF, which promote neurogenesis, synaptic plasticity, and mitochondrial biogenesis within neurons. Intermittent fasting and time-restricted feeding activate cellular stress response pathways that improve mitochondrial efficiency, support energy homeostasis, and promote neuronal repair. Controlled heat or cold exposure similarly induces adaptive mitochondrial responses, enhancing the brain's energy capacity and resilience. Sleep and circadian alignment also play a critical role in brain mitochondrial health. During deep sleep stages, neurons engage in repair, detoxification, and renewal processes that maintain mitochondrial function. Proper sleep allows for mitophagy, the selective removal of damaged mitochondria, and mitochondrial biogenesis, ensuring that neurons remain efficient and resilient. Disruptions in sleep compromise these repair mechanisms, leading to reduced energy availability, cognitive fatigue, and diminished mental performance. Prioritizing high-quality sleep strengthens mitochondrial function and supports sustained cognitive energy. Optimal mitochondrial function supports not only physical energy but also mood, stress resilience, and cognitive clarity. By maintaining efficient energy production, reducing oxidative stress, and promoting neuronal repair, mitochondria allow the brain to function at its highest capacity under both acute and chronic demands. Supporting mitochondrial health through exercise, nutrition, sleep, and hormetic stressors enhances brain energy, cognitive performance, and mental resilience. These strategies improve focus, memory, decision-making, and stress management while protecting neurons from oxidative damage, ultimately contributing to long-term mental vitality and overall longevity. Consistency, integration, and long-term lifestyle strategies are essential for maximizing mitochondrial health and sustaining energy and vitality. While individual interventions, such as exercise, nutrition, sleep, or hormetic stressors, can each provide measurable benefits, the cumulative effect of integrating these practices consistently is what drives profound and lasting mitochondrial adaptations. Mitochondria respond to repeated, structured challenges by improving efficiency, increasing in number, and enhancing their resilience to stress. Sporadic or uncoordinated efforts may produce short-term effects, but sustained intentional implementation is required to achieve optimal energy production, cellular protection, and long-term health outcomes. Regular exercise serves as a foundational pillar for mitochondrial optimization. Engaging in a combination of endurance, resistance, and high-intensity interval training ensures that mitochondria across multiple tissues are stimulated to grow, adapt, and function efficiently. Endurance training promotes mitochondrial density in slow-twitch muscle fibers, supporting sustained aerobic energy production. Resistance training targets fast-twitch fibers, improving energy utilization for short bursts of effort and increasing overall metabolic capacity. High-intensity interval training challenges both anaerobic and aerobic pathways, creating robust signaling for mitochondrial biogenesis. When these forms of exercise are practiced consistently, the body maintains a high baseline level of mitochondrial function and energy availability, enhancing both performance and resilience. Nutrition complements exercise by supplying the raw materials and cofactors required for optimal mitochondrial function. Nutrient-dense foods rich in B vitamins, magnesium, antioxidants, and healthy fats provide the essential components for ATP production, oxidative balance, and membrane integrity. Strategic dietary patterns, including intermittent fasting or time-restricted feeding, further enhance mitochondrial efficiency by activating adaptive stress response pathways and promoting mitophagy, the selective removal of damaged mitochondria. Over time, combining consistent nutrient support with regular metabolic challenges allows mitochondria to operate at peak capacity while reducing oxidative stress and cellular wear. Sleep and circadian alignment are critical for maintaining long-term mitochondrial health. Mitochondrial repair, mitophagy, and biogenesis predominantly occur during deep sleep stages. Consistently prioritizing high-quality sleep enables these restorative processes to happen efficiently, ensuring that energy production remains optimal and cellular stress is minimized. Aligning sleep-wake cycles with natural light-dark rhythms reinforces circadian regulation of mitochondrial enzymes and metabolic pathways, further supporting cellular energy efficiency and overall resilience. Without consistent sleep, even the most rigorous exercise and nutritional strategies cannot fully optimize mitochondrial function. Controlled environmental stressors, such as heat, cold, or brief periods of caloric restriction, serve as additional tools for mitochondrial conditioning. These hormetic challenges, when applied regularly and gradually, stimulate adaptive mitochondrial responses, enhance antioxidant capacity, and improve energy efficiency. Over time, mitochondria become more resilient to both metabolic and environmental stress, which translates into improved energy, recovery, and longevity. Consistency in exposure ensures that these adaptive mechanisms are reliably activated and reinforced. Integration of these interventions amplifies their benefits. Exercise, nutrition, sleep, and hormetic stressors work synergistically to enhance mitochondrial number, efficiency, and resilience. By consistently applying these strategies, the body maintains an optimized internal environment that supports energy production, metabolic flexibility, cognitive function, and overall health. Long-term commitment is essential, as mitochondrial adaptations develop gradually and are reinforced through repeated practice. By consistently combining exercise, nutrient-rich diets, high-quality sleep, and controlled hormetic stressors, mitochondrial function is maximized across multiple tissues and systems. This integrated long-term approach strengthens cellular energy production, enhances resilience to stress, and supports sustained vitality, mental clarity, and longevity, creating a FOW by committing to a consistent, integrated approach that combines exercise, proper nutrition, restorative sleep, and controlled hormetic stressors. The body is able to optimize mitochondrial function at a cellular level. Over time, these practices reinforce energy efficiency, enhance resilience to stress, support cognitive and physical performance, and protect against age-related decline. This long-term, lifestyle-based strategy provides a foundation for sustained vitality, mental clarity, and overall longevity, allowing individuals to maintain high-functioning energy and health throughout life. Foundation for optimal health throughout life.